Types of RNA. RNA is synthesized from DNA by an enzyme known as RNA polymerase during a process called transcription. Studies carried out in 1950s uncovered the relationship between genetic information and amino acid sequences. But this knowledge by itself didn't provide any clue to the mechanism by which the specific polypeptide chain is generated. As we now know, there is an intermediate between a gene and its polypeptide. The intermediate is a messenger RNA, that is mRNA. The discovery of mRNA was made in 1961. Francois Jacob and Jacques Monod of the Pasteur Institute in Paris, Sidney Brenner of the University of Cambridge, and Matthew Michelson of the California Institute of Technology. They showed that when a virus attacks a bacterial cell, a virus-specific RNA was made that is rapidly associated with the pre-existing bacterial ribosomes. Bacterial ribosomes were normal and they contained bacterial rRNA, but the new viral RNA was not a permanent part of the ribosome, but only transiently associated with them. Three types of cellular RNA are there, and all the major types of eukaryotic RNAs, that is mRNAs, rRNAs and tRNAs, are derived from precursor RNA molecules that are considerably longer than the final RNA product. The initial precursor RNA is equivalent in length to the full length of the DNA transcribed and is called the primary transcript or pre-RNA. The final product, that is mature mRNA, is obtained only after several modifications that will be discussed later. Classes of RNAs. First one, messenger RNA or mRNA or template RNA. Jacob and Monod in 1961 proposed the name messenger RNA for the RNA carrying information for protein synthesis from the DNA, that is genes, to the sites of protein formation, that is ribosomes. It consists of 3 to 5% of the total cellular RNA. Messenger RNA is the intermediary molecule that carries information from the DNA to the protein machinery so that it can be translated to functional protein. So, the mRNA is complementary to the gene from which it got transcribed. For this reason, mRNA can be described as sense strand or a coding RNA. Now, the size. The molecular weight of an average sized mRNA molecule is about 500,000 and its elimination coefficient is 8s. It should be noted, however, that mRNA varies greatly in length and molecular weight. Since most proteins contain at least 100 amino acid residues, mRNA must have at least 10 into 3, that is 300 nucleotides on the basis of the triplet code. Now, the stability of mRNA. The cell does not contain large quantities of mRNA. This is because mRNA, unlike other RNAs, is constantly undergoing breakdown. It is broken down to its constituent ribonucleotides by ribonucleases. Now, the structure of mRNA. Messenger RNA is always single-stranded. Since mRNA is transcribed from DNA, its base sequence is complementary to that of the segment of DNA from it got transcribed. This can be demonstrated by hybridization experiments with DNA and artificial RNA. Random coiling has been observed in extracted RNA, but there is no base pairing. In fact, base pairing destroys its biological activity. Usually contains the bases adenine, guanine, cytosine and uracil and sometimes have unusual substituted bases. Usually each gene transcribes its own mRNA. So, there might be 1000 to 10,000 different species of mRNA in a cell. These mRNA types differ in their base sequence and in length. When one gene, that is a cistron, codes for a single mRNA strand, the mRNA is said to be monocistronic. In many cases, however, several adjacent cistrons may transcribe an mRNA molecule, which is then said to be polycistronic or polygenic. The mRNA molecule has the following structural features. They contain a continuous sequence of nucleotides encoding a specific polypeptide. They are found in cytoplasm. They are attached to ribosomes when they are translated. Most mRNAs contains a significant non-coding segment, that is, a portion that does not direct the assembly of amino acids. Non-coding portions are found in both 5' and 3' ends of a messenger RNA and contain sequences that have important regulatory roles. These modifications include a 5' methylated cap, that is 7-methyl-guanosine cap, and a string of 50 to 250 adenosine residues that form a poly-A tail. The cap is followed by a region of 10 to 100 nucleotides known as non-coding 1 or NC1, rich in A and U residues and does not translate protein. The rate of protein synthesis depends upon the presence of the cap, without which the mRNA molecule poorly binds to ribosomes. Now, ribosomal RNA or rRNA. Ribosomes contain both protein and RNA. RNA found in ribosomes are called ribosomal RNA or rRNA. The eukaryotic ribosomes are larger than prokaryotic ribosomes. The 40S subunit of eukaryotic ribosome contains one rRNA, that is 18S rRNA, which is about 1900 bases and about 35 different proteins. The 60S ribosomes contains 3 rRNAs, 5S rRNA, it is about 120 bases, 5.8S rRNA, that is 160 bases, and 28S rRNA, about 4700 bases, and about 15 proteins. Like mRNA, each is transcribed from one of the DNA strands of a gene. 5S rRNA has its own gene. The others are synthesized as a single transcript that is then cleaved to release the mature RNA molecules that become part of the ribosome. Rather than functioning as a messenger, rRNA provides structural support and catalysis chemical reaction in which amino acids are covalently linked to one another. Ribosomal RNA consists of a single strand twisted upon itself in some regions. It has helical regions connected by intervening single strand regions. In the helical region, most of the base pairs are complementary and are joined by hydrogen bonds. Now, the soluble RNA, sRNA or transfer RNA or tRNA. Transfer RNAs are L-shaped molecules about 80 nucleotide in length. The shape is formed because of the complementarity between the bases. A typical tRNA has 4 short base paired stems and 3 loops. This is best explained in the clover leaf model proposed by Hawley in 1965. Different tRNA molecules vary considerably in sequence, but they all conform to the same overall structure. Variation length, that is about 73 to 93 nucleotides, occur, mostly due to the variable loop. Transfer RNA or tRNA is an essential component of the protein synthesis reaction. After rRNA, the second most common RNA in the cell is transfer RNA or tRNA. It constitutes about 10 to 20% of the total RNA of the cell. It is also called soluble RNA because it is too small to be precipitated by ultracentrifugation at 100,000 G. It got a molecular weight of about 25,000 to 30,000 and the sedimentation coefficient of a mature eukaryotic tRNA is 3.8S. Transfer RNA is synthesized in the nucleus on a DNA template. Only 0.025% of DNA codes for tRNA. Synthesis of tRNA occurs near the end of cleavage stages. The function of tRNA is to carry amino acids to mRNA during protein synthesis, translating the information in the mRNA nucleotide codon into amino acid alphabet of a polypeptide. In fact, tRNAs act as an adapter molecules, that is, each tRNA is linked to a specific amino acid as an amino acid tRNA, that is AAtRNA. And while on the other hand, the same tRNA can recognize a particular codon in the mRNA. The interaction between successive codons in the mRNA and specific amino acid tRNAs leads to the synthesis of a polypeptide with an ordered sequence of amino acids. Unusual bases in tRNA. In addition to the usual bases A, U, G and C, tRNA contains a number of unusual bases and in this respect differs from mRNA and rRNA. Most of the unusual bases are formed by methylation, addition of CHA or methyl group to the usual bases. Example, cytosine and guanine on methylation yields methyl cytosine and methyl guanine respectively. Precursor tRNA molecules transcribed on the DNA template contains the usual bases. The unusual bases are important because they protect the tRNA molecule against degradation by RNAs. This protection is necessary because RNA is found floating freely in the cell. Some of the unusual bases of tRNA are methylguanine GME, dimethylguanine GME2, methylcytosine ME, ribothiamine T, pseudouridine Psi, dihydrouridine DHU, H2U, UH2, then inosine I and methylinosine IME or MEI. In general, organisms high in the evolutionary scale contain more modified bases than lower organisms.